So what are the two types of nucleic acids you come across in the nucleus of a cell? DNA and RNA. So usually in 3-marker question and 5-marker question, split questions might be asked. As I keep telling you all, for 3-marker it might be 2 plus 1, or for 5-marker it might be 3 plus 2 or 2 plus 2 plus 1 also. What is nucleotide? Nucleoside plus phosphate makes a nucleotide. So what is the bond here? It is phosphodiester bond. Hello everyone, welcome back to this revision session of 2nd PUC, chapter 5 called Molecular Basis of Inheritance. Myself, B. Shobarani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. This particular chapter, chapter 5 of 2nd PUC Biology called Molecular Basis of Inheritance, has a weightage of 14 marks. So it's a very big marks, and the concepts what you study from this chapter are very, very important from examination point of view. So let us discuss about the important concepts that are related to this particular chapter called Molecular Basis of Inheritance. Moving on to the flow chart, experiments for genetic material. Search for genetic material. DNA is called as the genetic material. Search for genetic material was done by Griff, so it is called Griff's experiment, then Hershey-Chase experiment, and biochemical characterization of transforming principle. Then structure of a polynucleotide chain. As we know that DNA is an organic macromolecule found in the nucleus of a cell and shows acidic property. So what are the two types of nucleic acids you come across in the nucleus of a cell? DNA and RNA. So here, what is the chemical composition of DNA and RNA? How does DNA differ from RNA? So DNA and RNA both are made up of nitrogenous bases, that is purines and pyrimidines, that is adenine, guanine, cytosine and thymine, and a pentose sugar that is ribose sugar and deoxyribose sugar, and a phosphate group. So only when a phosphate group is added, you call it as nucleotide. Then discoveries with respect to structure and composition of DNA was done by Frederick Mister. The word nuclein came into existence, it is because of Frederick Mister. Then James Watson and Francis Crick, who proposed the double helical model of DNA. Then Erwin Chargaff. So nuclein and double helix model of DNA was put across by James D. Watson and Francis H.C. Crick. Then Erwin Chargaff's rule is that purines never base pair with purines only, nor pyrimidines with pyrimidines. Purines base pair with pyrimidines and pyrimidines base pair with purines. Erwin Chargaff's rule, A plus T and G plus C is equal to 1. Then enzymes involved in DNA replication, that is DNA polymerase, helicase and DNA ligase. Transcription unit, the flow chart of transcription unit includes a promoter, a structural gene and a terminator. Then types of RNA, as we all know that RNA acts as both as genetic RNA and non-genetic RNA. So when RNA is acting as a genetic RNA, it is only single-stranded, but when it is acting as non-genetic RNA, there are 3 types of non-genetic RNA, mRNA which is called messenger RNA, rRNA which is called as ribosomal RNA, and tRNA that is called as transfer RNA. Then proposal for genetic code, George Gamow put across 3 nucleotides should make up the code, that is a triplet codon system was introduced by George Gamow. Hargobind Khurana, chemical method for synthesizing RNA with base. Marshall Nirenberg, cell-free system. Then Severo Okawa, enzymatic RNA synthesis. Then structure of tRNA. As we know, tRNA is also called as adapter molecule. So it has the cloverleaf model of tRNA includes the anticodon loop, amino acid acceptor, T loop, D loop and variable loop. Then stages of protein synthesis includes initiation, activation of amino acid, transfer of amino acid to tRNA, elongation of polypeptide and termination of polypeptide. Then regulation of gene expression, which is also explained through transcription level, processing level, transportation level and translational level. Then steps in DNA fingerprinting includes DNA isolation or separation, amplification and digestion of DNA. Moving on to the MCQs, that is the multiple choice questions. Here in this particular revision class, I have given one example to make you understand what type of multiple choice questions appear from this particular chapter, chapter 5 of your second PUC biology, called Molecular Basis of Inheritance. As I said in the beginning of my classes itself, that multiple choice questions are very, very important for you people to, particularly for the second PUC students to score. So one example I have given, the first genetic material could be either is it a protein or carbohydrate, DNA and RNA. So the first genetic material was what? RNA. So the answer is option D. Moving on to the next question, write the two basic amino acid residues which are rich in histones. The answer is lysine and arginine. Here I have come across the questions which were repeatedly asked in the previous question papers of the PU board examination. So mention any three applications of DNA fingerprinting technique. It is applied in forensic science to identify the culprits or the criminals who have committed the crime, in determining population diversities, evolutionary biology, and in determining genetic diversities. Question number 4. Describe Frederick Gift experiment to show transformation in bacteria. So this is a very important question from examination point of view, which is asked for 5-marker question. So here, what is this all experiment? So this experiment proves that DNA is a genetic material. So what did Griffith do? He took mice infected with the S strain, smooth strain of Streptococcus pneumoniae, which was virulent, died from pneumonia infection. But mice infected with the R strain, that is the rough strain, did not develop pneumonia and were alive. Next step, what did he do? Next, he took the mice infected with heat-killed S strain bacteria and it did not kill them and the mice were alive. So, next he took what? Heat-killed S strain. Next step, when he injected a mixture of heat-killed S strain and live R strain bacteria, the mice died. Moreover, he recovered what? Living S bacteria from the dead mice. He concluded, so with this simple experiment, what did he conclude? He concluded that the R strain, that is the rough strain bacteria, had somehow been transferred. Actually, R bacteria was non-virulent. How was it transformed into virulent? By the heat-killed S strain bacteria. There is some transforming principle which transformed R bacteria into S bacteria, thereby killing the mice. So, some transforming principle transferred from the heat-killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. So, from S, the genetic material, that is the smooth bacteria, the genetic material was transferred to R strain and that is how it killed the mice. So this is a very important concept from examination point of view. Moving on to the question number 5. Differentiate between linkage and recombination. This is also a 2-marker question which is asked. Linkage. What is the difference between linkage and recombination? Linkage, physical association of the genes on a chromosome and inherit together without segregation is called linkage. Then what is recombination? The generation of non-parental gene combination is called recombination. Next, question number 6. Write any 5 salient features of genetics. So this is usually for 5-marker question. This is also very, very important question, which is usually, you might have just gone through the previous question papers. This is one such question which is repeatedly asked. So this is very important from examination point of view. So what are the salient features? The codon is triplet. As we know, the codon system what we are following right now is a triplet codon system. What is triplet codon system? Usage of 3 nitrogenous bases coding for a particular amino acid. So actually there are 64 codons are there, but 61 codes for amino acids and remaining 3 does not code for any amino acids, where as such they are called as what? They are called as stop codons, or also they are called as what? Nonsense codons. What do you mean by nonsense? The codons which make no sense, right? Next, One codon codes for only one amino acid. Hence, it is called as what? Unambiguous and specific. They are very specific. Some amino acids are coded by more than one codon. Hence, the code is degenerate. The codon is read in mRNA is a continuous fashion. There are no punctuations. The code is nearly universal. The code is nearly universal. For example, from bacteria to human, UUU would code for what? Phenylalanine, an amino acid called phenylalanine. So here only you can see UUU is a triplet codon. Usage of 3 nitrogenous bases. Usage of 3 nitrogenous bases. Then AUG has dual function. AUG has dual function. What is it? It performs 2 very important functions. AUG codes for a very important amino acid called methionine, and it also acts as an initiator codon. That is, protein synthesis always starts with AUG. So AUG has dual function. It codes for methionine and it is also act as an initiator. So always the protein synthesis starts with the AUG and ends with the stop codons or nonsense codons. Moving on to the question number 7. This is also very important from examination point of view, which is asked for 5-mark question. As I said, this chapter called Molecular Basis of Inheritance, chapter 5 of 2nd PUC Biology, has a weightage of 14 marks. So this is very important chapter from examination point of view. So you must make sure that you study the concepts of this particular chapter thoroughly. Describe the Hershey-Chase experiment. This is called Hershey-Chase experiment, done to prove what? That DNA is a genetic material. So what did they do? Hershey and Chase grew some bacteriophage. What is bacteriophage? Virus which infects a bacterial cell is called as a bacteriophage. Viruses on a medium containing radioactive phosphorus. So P32, phosphorus 32, and some others on a medium containing radioactive sulfur, that is S35. So what did he do? Hershey and Chase grew some bacteriophage viruses on a medium containing what? Radioactive phosphorus, and some others on a medium containing sulfur, radioactive sulfur. Then viruses grown in phosphorus, that is radioactive phosphorus 32, got radioactive DNA because only DNA contains phosphorus. As we all know, DNA is made up of 3 very important chemical composition: a nitrogenous base, a pentose sugar, and a phosphate group. So obviously, viruses grown in radioactive phosphorus 32 contain only DNA because DNA contains what? Phosphorus. Viruses grown in sulfur, radioactive sulfur 35, got radioactive protein because protein contains sulfur. Next, these preparations were used separately to infect E. coli, Escherichia coli. After infection, the E. coli cells were gently agitated in a blender to remove the virus particles from the bacteria. Then the culture was centrifuged to separate lighter virus particles from heavier bacterial cells. Next step, bacteria infected with viruses having radioactive DNA were radioactive, that is DNA had passed from the viruses to the bacteria. Bacteria infected with viruses having radioactive proteins were not radioactive. Why? Because proteins did not enter the bacteria from the viruses. So with all this experiment, what did Hershey and Chase try to prove? Prove that DNA is the genetic material. So this particular question is very important from examination point of view, and there is a very beautiful diagram given in the NCERT textbook. Please go through with related to this Hershey-Chase experiment. Moving on to question number 8. Name the pyrimidine base present both in RNA and DNA, that is cytosine. Next, 9. Write the differences between euchromatin and heterochromatin. It's a split question. Name the virus used in Hershey-Chase experiment. What is the difference between euchromatin and heterochromatin? In a typical nucleus, what is euchromatin? Loosely coiled regions of chromatin with light stain and transcriptionally active is called euchromatin. So loosely coiled regions of chromatin with light stain and transcriptionally active is called euchromatin. The chromatin that is densely packed, stains darker, and transcriptionally inactive is called heterochromatin. So this is a very important question for 2-mark question. Differentiate between euchromatin and heterochromatin. Next split question. Name the virus used in the Hershey Chase. So this is a 3-marker question. So write the difference between euchromatin and heterochromatin is 2-marker question, and name the virus used in Hershey Chase experiment is 1-mark question, that is bacteriophage. So usually in 3-marker question and 5-marker question, split questions might be asked. As I keep telling you all, for 3-marker it might be 2 plus 1, or for 5-marker it might be 3 plus 2 or 2 plus 2 plus 1 also. So this is one such question, split question, which is asked for 3-marker question. Moving on to question number 10. Mention the steps involved in DNA fingerprinting. Already I had shown this in the flow chart. So what is the first steps? Isolation of DNA, digestion of DNA by restriction endonucleases, separation of DNA fragments by gel electrophoresis, transferring, that is blotting of separate DNA fragments to synthetic membranes such as nitrocellulose or nylon, detection of hybridized DNA fragments by autoradiography. This is also very important question from examination point of view. This is very important. Next, name the inducer which regulates the switching on and off of the lactoparent concept. As you know, lac stands for what? Lactose. It is lactose. Then, which type of RNA polymerase enzyme transcribes precursor mRNA? It is RNA polymerase 2. Then, 13th question. Name the scientist who found out DNA and what was the name given by him? Who discovered? It was Frederick Mister, and the name given by him was nuclein. So, Frederick Mister subjected the hospital bandage pus cells with certain digestive enzymes, and one part of the cell remained undigested, and that is how he called that particular part as nuclein. So he was the person who coined the word called nuclein, that is nucleic acid. Because why the name nucleic acid has come? They are found in the nucleus of a cell, and they are organic macromolecules found in the nucleus of a cell and show acidic property. Next, mention 5 salient features of HGP, that is Human Genome Project. This is also very important from examination point. Usually they ask this question in the question paper. Human genome contains what? 3,164.7 million nucleotide bases. Total number of genes are about 30,000. Average gene consists of 3,000 bases, but sizes vary. Largest known human gene, dystrophin on X chromosome, contains 2.4 million bases, and about 99.9% nucleotide bases are identical in all people. It is that 0.1% is what makes each one unique. Functions of over 50 of discovered genes are unknown. Chromosome 1 has most genes and Y has the least genes. How much? 2,968, and it has Y gene has what? 231. Repetitive sequences are stretched of DNA sequences that are repeated many times. They mark the largest portion of DNA. They have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. There are about 1.4 million locations where single base DNA differences, that is SNP means single nucleotide polymorphism, occurs in human. Moving on to the question number 15. Codon AUG has dual function. Give reason. This is a 2-marker question. So I have in the previous slide I explained what are the salient features of genetic code. In that, one salient feature is what? AUG has dual functions. So the acts as what? Initial codon and also codes for an amino acid called methionine. Explain biochemical characterization of transforming principle of Oswald-Avery. This is also very important question from examination point of view, which is for a 5-marker question. So what is this biochemical characterization of transforming principle? Biochemical nature of transforming principle was discovered by whom? Oswald-Avery. Hence it is also called Oswald-Avery transforming principle. Then MacLeod and MacLean-McCarthy in 1933 to 1944. Prior to their work, genetic material was thought to be a protein, but they worked to determine the biochemical nature of the transforming principle of Griff's experiment. Already we have, in the previous slide, I explained about Griff's experiment, which shows the DNA is a transforming principle. They purified biomolecules. What are they? Proteins, DNA and RNA from the heat-killed S cells to see which one could transform live R cells to S cells. Heat kills S strain plus protease plus live R strain, there was transformation. Heat kill S strain RNase plus live R strain, transformation. But heat kill S strain DNase Live R strain, no transformation. So this is also a very important question from examination point of view. Then describe lack apparent concept of gene regulation, that is switch off and switch on mechanism. This is also very important question from examination point of view for 5-marker question. So when lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. The regulator gene produces a kind of protein called repressor that binds to the operator gene. This do not allow RNA polymerase enzyme to transcribe the structural genes. Hence, there is no transcription, translation, and the required protein is not synthesis. So you call this whole mechanism as what? Switch off mechanism. Like how we switch on the lights when we want, and when we don't want the light, we switch off. So same mechanism, switch on mechanism. When the lactose molecules, so when the lactose molecules are not present, switch off. When it is present, lactose molecules are present in the nutrient medium, DNA is switched on for protein synthesis. The lactose molecules added in the nutrient medium acts as what? Inducer, which binds to the repressor to form inducer-repressor complex. And now the repressor become inactive and release from the operator gene. This enables the RNA polymerase enzyme to transcribe all the 3 types of structural genes. What are they? Z, Y, and A. This results in the synthesis of 3 mRNA molecule continuously and are called polycistronic mRNA. All these 3 mRNA, there is also a diagram depicting the switch on and switch off mechanism, which is given even in the NCERT textbook, which is very important for you all. When you explain this concept, please do explain with the diagram, schematic diagram what is given there. All these 3 mRNA strands undergo translation to produce 3 polypeptide chains or 3 proteins which act as 3 enzymes such as beta-galactosidase, permease, and transacetylase. These 3 enzymes together break lactose into glucose and galactose. In polycistronic mRNA, the product of the gene activates the second gene, the product of the second gene activates the third gene, and so on. So this is called pre-programmed circuits of gene action. So this whole concept of lack apparent concept is very important from examination point of view, which appears for 3-marker question. Moving on to the next question. Which chromosome of man has the least number of genes? Chromosome 1 has highest number of genes, and Y chromosome has least, that is 231. So it is Y chromosome. Question number 19. DNA is the better genetic material than RNA. Justify the statement with 3 comparative reasons. This is very important question, which is usually asked for 3-marker question. So you have to give 3 reasons why DNA is a better genetic material than RNA. DNA, that is structural and chemically, DNA is more stable than RNA. DNA mutates slow, but RNA mutates faster because of the hydroxyl group in RNA is a reactive group and makes RNA liable and easily degradable. DNA is less reactive than RNA. Presence of thymine at the place of uracil give additional stability to DNA. Adenine, guanine, cytosine, and thymine is present in DNA. Whereas in RNA, what are the 4 nitrogenous bases? Adenine, guanine, cytosine, and uracil. So the thymine of DNA is replaced by uracil. That is the reason why thymine at the place of uracil gives what? Additional stability to DNA. Hence, DNA acts as a better genetic material than RNA. Next, DNA replication is said to be conservative. This is also very important question from examination point of view. DNA replication is said to be semi-conservative. What do you mean by semi-conservative? Half of the DNA parental strand is retained in the daughter cell, hence it is called semi. Semi means half of the parental strand is conserved or preserved in the daughter cell. So why it is called semi-conservative? Describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. So why DNA replication is said to be semi-conservative? In both the daughter DNA molecules, one strand is parental, which is otherwise called as what? Template strand. Template strand, and another is newly synthesized one. Hence, DNA replication is called as what? Semi-conservative. It was first shown by whom? Matthew, Michelson, and Franklin Stahl in 1958 in E. coli, and subsequently in higher organism. It is popularly known as what? Michelson and Stahl experiment. They grew E. coli in 14, that is ammonium chloride medium. Ammonium chloride medium for many generations. As we know, 15N is heavy isotope of nitrogen. The result was that the 15N was incorporated into newly synthesized DNA and other nitrogen containing compound as well. This heavy DNA molecule could be distinguished from normal DNA by centrifugation in cesium chloride density gradient. Then they transferred the E. coli into a medium with normal, that is 14NHCl, and let them grow. So here what happens, E. coli divides in 20 minutes. They took samples of definite, that is time intervals as the cells multiplied, and extracted the DNA that remained as double-stranded helices. So various samples were separated independently on cesium chloride gradients to measure the densities of DNA. The DNA that was extracted from the culture, that is one generation of the other, transfer from the 15N to 14N medium, a hybrid or intermediate density. So DNA evidently that the culture after another generation was composed of equal amount of this hybrid DNA of light DNA. So moving on to question number 21. List the goals of Human Genome Project. There in the previous slide I explained about the features of Human Genome Project. Here the goals, the aims of HGP, very important, which is asked for 2-marker question. So you have to give 4 goals. So it is used to identify all the approximately 20,000 to 25,000 genes in human DNA, determine the sequence of all 3 billion chemical base pairs, store this information in databases, improve tools for data analysis, transfer related technologies to other sectors such as industries, address the ethical, legal and social issues. Put together it is called as ELSI that may arise from the project. This is very important question, 2-marker question from examination point of view. Moving on to question number 22nd. Mention the criteria for a molecule that acts as a genetic chemist. This is also very, very important, which is asked for in the previous years they have asked this question. So it is very, very important. So what are the 3 criteria for a molecule that acts as a genetic? It might be asked for 3-marker question. Or see here it is split 2 plus 1. So it should be able to generate its replica. Then it should be chemically and structurally stable. It should provide the scope for slow changes or mutation that required for evolution. It should be able to express itself in the form of Mendelian character. So what are the 3 criteria for a molecule to act as a genetic material? What is it? It should be able to generate its replica. It should be chemically and structurally stable. It should provide the scope for slow changes that required for evolution. It should be able to express itself in the form of Mendelian characters. Moving on to question number 23. Again, it is a split question. Mention the chemical linkages between the components of DNA. So, nitrogen. So, we all know the 3 chemical composition of DNA is what? 3 nitrogenous bases, that is the 2 nitrogenous bases. Like we come across 4 nitrogenous bases, then a pentose sugar, that is a deoxyribose sugar, as well a phosphate group. So what is the linkage present between the components of DNA? Differentiate euchromatin from heterochromatin. Already I had explained this in the previous slide, but this is a question which is a split question which comes along with the split questions. So in between the nitrogenous bases, what is the bond we find? Hydrogen bond. In between the nitrogen base and sugar, glycosidic bond. In between the nucleotides, phosphodiester bond. This is very, very important point to be noted. The linkages between the components of DNA. So what is the type of linkage you come across between the nitrogenous bases? Hydrogen bond. And in between the nitrogenous bases and sugar, where we call it as nucleoside, it is glycosidic bond. Nucleotide is what? That is nucleotide means what? What is the bond between? What is nucleotide? Nucleoside plus phosphate makes a nucleotide. So what is the bond here? It is phosphodiester bond. Next, in a typical nucleus, some loose, I already explained euchromatin and heterochromatin. Euchromatin is what? Loosely coiled regions of chromatin with light chain, and transcriptionally active is called euchromatin. The chromatin that are most densely packed, stains darker and transcriptionally inactive is called heterochromatin. This is very important question. Codon AUG has dual function. Justify. Already I had explained this in the previous slide regarding the salient features of genetic code as well. AUG acts as a dual function. So one of the very important salient features of genetic code is what? Codon AUG has dual function. Justify. AUG has dual function. It is a triplet codon which codes for an amino acid called methionine. As well, it is an initiator codon. So any protein synthesis will always start with AUG, that is methionine, and always end with the stop codons or nonsense codons. What are the criteria of DNA to act as a genetic? Undergo replication, chemically, structurally, already I explained. So this is how, why? Because again I have explained here, it means to say that it has been repeatedly asked in the previous question papers. Chemically and structurally stable, slow changes, that is mutation that are required for evolution, able to express itself in the form of Mendelian characters. 26th question, list the salient features of double helix model of DNA. Here you can see the beautiful picture of double helix model which was put across by Watson and Crick model. So it is called Watson and Crick double helical model. So what is the feature of this double helical structure of DNA? Here you can see here it is 20 Armstrong units and the length is what? 3.4 nm nanometer, and the internucleotide distance is 0.34. And here you can see the minor groove and the major groove and the hydrogen bonds that are found between the nitrogenous bases. So based on the observations of Morris, Wilkins and Rosalind Franklin's X-ray diffraction studies, Watson and Crick came across with a model to explain the structure of DNA which is called double helical model, for which they were awarded the Nobel Prize. So James Watson and Francis Crick proposed double helix model for the structure of DNA in 1953 and published the discovery in a scientific journal called Nature. What are the features? According to this model, DNA is composed of two polynucleotide chains which are oriented in anti-parallel manner, that is 5', 3', 3' and 5'. The two chains are coiled in a right-handed fashion. The polynucleotide chain on one strand is formed by phosphodiester linkage in 5', 3' direction, whereas the phosphodiester linkage on other strand is 3', 5'. As I already said, it is a double-stranded structure and it is anti-parallel. Hence, both these DNA strands are said to be arranged in an anti-parallel manner. Both the polynucleotide chains are held together by means of hydrogen bonds formed between nitrogen bases. Here, adenine pairs with thymine. As I already said, Erwin-Chargaff's rule is what? A base pairs with double bond with T, C with triple bond with G. So, A is stable with thymine with two double bonds, that is hydrogen bonds. Here, that is cytosine is stable base pairs with guanine with 3 hydrogen bonds, with thymine by 2 hydrogen bonds, and guanine pairs with cytosine by 3 hydrogen bonds, that is 3 hydrogen bond. Hence, DNA, in a DNA molecule, the number of purines is equal to number of pyrimidines, and this is called as what? Erwin-Chargaff's rule. Then, both the polynucleotide chains take a turn for every 34 Armstrong units, that is 3.4 nm, that contain roughly 10 years. So, one twist or one helix, I think the previous diagram you can see, one twist or one helix will have around 10 nucleotides. Then, hence the internucleotide distance is what? 3.4 Armstrong. So, here you can see the internucleotide distance is what? 0.34 nm. Then the diameter of dimension of two polynucleotide chains is about 20 Armstrong. So, the diameter is 20 Armstrong. In a DNA molecule, the pentose sugar and phosphate form the backbone of DNA. So, totally what are the three very important chemical composition of DNA molecule? 4 nitrogenous bases, that is adenine, guanine, cytosine and thymine, and pentose sugar called deoxyribose sugar and a phosphate group. Moving on to the next question, 27th question. tRNA is an adapter molecule. Comment. This is asked for 2-mark question. It is an RNA molecule that transfers amino acids to the site of protein synthesis, that is into the ribosome during protein synthesis, hence the name adapter. So, this is the structure of the adapter molecule, tRNA molecule with having anticodon loop. Here you can see amino acid binding loop. So, this is the adapter molecule. Moving on to question number 28. Describe the process of DNA replication with the help of a diagram. This is also a very important question from examination point of view, which is asked for 5-marker question. So, this is important. Please make note that this chapter has lot of weightage of marks of that too of 14 marks. So you can understand how important the concepts that you are going to study, because maximum marks, I told you already, principles of inheritance also 14 marks, and this chapter also. So these two are such chapters which have lot of weightage of marks. So you need to concentrate more on these two chapters where it will be easy for you all to score. DNA replication, the machinery and the enzymes. So the parental DNA molecule produces two identical copies of daughter DNA molecules, is called DNA replication, and it takes place in the nucleus of a cell before cell division, that is in the S phase of interphase stage. As we know, in the S phase of interphase, DNA replication starts. The process of replication begins at a specific site. What do you call that site as? ORI, origin site. Then the double helix starts unwinding, or you can also call it as unzipping from the origin, catalyzed by unwinding proteins and helicase enzymes. The hydrogen bonds present between two strands are dissolved by DNA polymerase 1 and topoisomerases, that is DNA gyrases. Next, what happens? The double-stranded DNA is now getting separated. So here we know, like, it starts separating like this. This mechanism is called unzipping of DNA. So it is like, before it is winded like this. Later on, it unwinds like this. As the double helix separated, it gets into Y shape. So you can also call it as Y fork. In the previous slide, I am going to show you all the diagram. Y-shaped appearance called replication fork. Both the separated single-stranded DNA access templates on which new strands are going to be seen. For example, if this is one, this is called as a template strand. And this is also called as a template strand. For this, the newly synthesized strand is going to form. For example, here if it is A, T, C, G, T, A. So here it will be what? A, G, T, C. For this, A with T, G with C, T with A, C with G. So like this. Next, before the addition of DNA nucleotides onto the template, a short segment of tRNA strand is synthesized called RNA primer, catalyzed by RNA primase enzyme. The length of RNA primer is maximum of 10 nucleotides. Further addition of DNA nucleotides takes place to the exposed bases on the templates continuously towards the replication fork from the end of RNA primer is called the leading strand. Here you can see the diagram of, and this is called as replication fork diagram. So very, very important from examination point of view. So here you can see Okazaki fragments, the leading strand and the lagging strand, DNA ligase, DNA template strand. So this diagram is very, very important from examination. So you need to explain the concept of DNA replication with this diagram called replication fork. The Okazaki fragments are joined by DNA ligase enzyme to form a lagging strand. DNA-dependent DNA polymerases catalyze polymerization of DNA nucleotides in a leading and lagging strand. Polymerization of new nucleotides takes place only in one direction, that is 5-3- direction. Here you can see 5-3- direction. The synthesis of DNA proceeds towards both the directions from the origin, hence it is called bidirectional. In both the daughter DNA molecules, one strand is parental and the other is newly synthesized. Because one of the parental strand is retained in the newly synthesized daughter molecule, hence it is called as semi-conservative. Next, question number 29. Lactose is termed as inducer, as we already studied in lactoparent concept. Why lactose is called as an inducer? Give reason. Because it regulates what? The switching on and switching off. Switching off is mainly due to absence of lactose. Switching on is due to presence of lactose. So it acts as an inducer, starter. So it regulates switching on and switching off of the operant genes. 30th question. Define transcription. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription, or there is one more definition for it. The synthesis of RNA from a template strand of DNA with the help of DNA-dependent RNA polymerase in the nucleus of a cell is called transcription. Hope you all have understood the important concepts what I have explained in this revision class of chapter 5 called Molecular Basis of Inheritance, which has a weightage of 14 marks. So all the questions what I have discussed in this particular revision class or revision session is important from examination point of view. So kindly go through the concepts and the type of questions that are going to be asked from this particular chapter called Molecular Basis of Inheritance. Thank you.